So it's not quite 9 o'clock, so I can see that we have some people joining, but bear with us for a few moments whilst people log in. I can see that some names are appearing in the list of attendees. If you can hear me, could you just type yes or hello or hi or something in the chat? Just to great, fantastic, thank you. It's always very reassuring when I know that everything's working, that you can hear us, and everything is in action. So that's really, really good. So whilst people are logging in and joining, I'll just ask a couple of questions just out of curiosity. So I'm in London at the moment for broadcasting this. I'm actually doing it from home rather than in the office. But could you just let us know in the chat if you're happy to? Just let us know where in the world you are. It's always really interesting to know where our audience is. It's 9 o'clock here in the UK, and it's fascinating to know. So somebody else is joining from London. It's first thing in the day in London. So thank you for joining us. Just on break. Someone from South Korea and Denmark. It's really interesting to know that we've got people from very different parts of Switzerland. So a couple of people from Europe, somebody from Indonesia. Very, very interesting. And someone from Japan. Well, a very warm welcome. Great to have you with us. Very interesting to know that at 9 o'clock in the morning, we've got people from UK, London, and then sort of further, quite far east. So that's very interesting for us. And thank you for letting us know. So it's 9:01. I can see that we've got a few people joining. I'm going to start explaining what this session is and how it's going to run. First of all, my name is Kim Morgan. I'm the Outreach and Communications Coordinator for UCL Biochemical Engineering, and it's an absolute privilege to welcome you for this, the second of our virtual taster lectures. So as you will have seen already, there's a chat function. If you look on your device, whatever you're using, whether you're using a desktop computer or a tablet or a mobile device, you actually have you have little options within Zoom. So you can use chat, Q&A, you can reconfigure the screen, whatever works for you, however you're configuring it. So you've got a few choices there. If you have any technical problems throughout this session, if you're having issues with people with hearing us or anything like that, please let myself and Travis know using the chat function. We'll get to see that straight away, and we'll try and sort out those problems. We're really looking forward to your questions. It's great. Yesterday we had some fantastic ones. I'm sure we're going to get some great ones today from you. Please use the Q&A function though. It really helps us out. It's a lot easier to see the questions as they come through. You can ask them at any point, but we'll come to them at the end. It's very useful if you ask them to use the Q&A function, because sometimes you get a lot of thematic questions. So it's easier to answer a few at a time. Or sometimes Travis will have actually addressed that during the session as well. So it really helps us. If you put it in the chat, sometimes they can get lost. So please use the Q&A function. You can also ask them anonymously if it's something that you really think you don't want to put your name to. It's absolutely fine. You know, you're welcome to ask any questions you want. So as I said, this session is being recorded. We'll make a link available afterwards to everyone who's registered for every one of these virtual taster lectures. So you'll be able to watch yesterday's one on stem cell gene therapies and cancer treatment, and the ones coming up on vaccines and so forth. You'll be able to see all of those. So one of the things that I just want to make clear is that if we touch on any scientific concepts that you're not familiar with, if you were to come and study a degree with us, we would take you to that level. So don't feel like, I don't understand this, I wouldn't be able to do a degree here. That's fine. We have modules designed to take you to the level of mathematics, or biology, or chemistry, whatever it is that we touch on here. So by all means, ask us to clarify a concept, but don't, you know, if we start to hit on some quite advanced things, that's fine. That's what we do at our universities. We teach that to you. Also just want to check, is there anything else I need to go through? So I think that's it for now. If I turn my screen off, oh yeah, sorry, before I do that as well, it'd be really good to know something about yourself. So I'm going to launch a little poll just to ask you whether you have an offer to study and what sort of level you would be interested in. So first of all, do you have an offer to study a degree with us here at UCL Biochemical Engineering or at UCL, or not at all? We've advertised this to people through a number of medium, and it's just really good to know whether people who have got an offer have come along today. So looking at the people who've responded, It looks like a fairly good split of people who have an offer to study with us, and people who don't. So that's really interesting. Thank you for filling that in. That's very helpful. And the next one we just want to ask is what sort of level you would be more interested in, whether you're considering doing a degree or a postgraduate degree like an MSc or an MRes or something. So at the moment, it's roughly split equally between prospective undergraduate and prospective degree students, which is really interesting. Thank you very much for telling us that. That's very helpful for us indeed. So thank you for taking a moment to fill that out. Right, so I'm going to hand over to my colleague Travis, and he will talk. I may turn my camera off, but I will be here in the background to deal with any questions you have. I'll also post some stuff in the chat, so I'll repeat all this. Thank you for bearing with me through the introduction. Travis, over to you.
Share my screen. You see this? Okay.
Yep, that's fine.
Yep, in the right mode.
At the moment, it's in presenter mode, and now it's in full screen. Thank you.
Perfect. Well, thank you all for coming. I'm Travis, so yeah, I'm here to talk about cultivated meat today. So I'll jump right into it. So who is this buffoon you see before you? Well, I'm Travis, and obviously I'm a postdoc at UCL. But 11 years ago, I was an undergrad graduating from Keele Uni with a degree in biomedical sciences, wondering what to do with my career. And since I care a lot about animal welfare, I decided to try and dedicate my career to that. So I embarked on a PhD in cultivated meat at the University of Bath. And after that, I worked for a couple of years at a cultivated meat startup in Cambridge. I don't have many photos from that, so here's one from a Halloween party. And now here I am as part of Petra's group at UCL. So Petra, who many of you might already have heard of, is here in the captain's chair. Postdocs include myself, of course, and Maria Laura. And we also have PhD researchers William, Juman, Joanna, Isa, who has a different supervisor, but a lot of overlap with us, and Helder. Great bunch of people, and I'm sure you'll be seeing them in the field quite soon. So what am I talking about? Cultivated meat, also known as cultured meat, clean meat, slaughter-free meat, cruelty-free meat, and when I first entered the field, it was in vitro meat. So whilst traditional meat involves raising an animal who is then killed and the meat removed for food, the cultivated meat approach involves starting with cell cultures from an animal from which meat is then grown and used for food without the animal being killed. And the most obvious benefit is to animal welfare. Whatever your stance on meat consumption is, we can hopefully all agree that factory farms and the conditions animals have to endure there need to be addressed. But with the population set to grow exponentially and the demand for meat with it, these problems are only going to get worse. So we really need an alternative. And we also need an alternative from an environmental standpoint. The meat industry is responsible for more greenhouse gas emissions than all forms of global transport combined. And conventional meat production uses up 1/3 of global ice-free land. So cultivated meat isn't just a single field, but rather an intersection between many different fields, any one of which you could devote an entire career to researching. And I'm going to give you a very quick introduction to them being cell sourcing, scaffold building, media development, and bioreactor design. So we'll start with cell sourcing. There are lots of different types of cells we can use here and lots of different ways of getting them, but I'm going to overview just two of them, myosatellite cells and induced pluripotent stem cells, or iPSCs. So let's start with myosatellite cells, which are the most used in the business. So for this, we need a small biopsy of muscle tissue, which we can get with a needle. Let's pretend this is a pig or some other farm animal. Now, I'm assuming most of you are familiar with the general structure of muscle being composed of bundles of contractile fibers. What you may not know about is the cells that hang out on them in a dormant state. These are myosatellite cells, and they're basically sleeping muscle progenitor cells. And when they detect damage to the muscles, such as from micro tears from lifting weights in the gym, they activate, wake up, and they start dividing to form new muscle tissue, repairing the damage and increasing the total mass of the muscle, which is how you get swole, as the young people say. We isolate some of those cells and encourage them to do their thing in a lab setting and make muscle tissue for us. Now, because these cells are isolated straight from living tissue, they're called primary cells. And the problem with primary cells, at least for our purposes, is that after a while, they get tired of dividing and they stop doing it, which means either we need a donor herd from which we can regularly get biopsies to replenish our cell supply, or we need to find a way of getting the cells to keep dividing for longer, which is the aim of iPSCs, induced pluripotent stem cells. And what we do here is take primary cells and do a little reprogramming to make them revert to a more stem-like state, which means they're more inclined to keep dividing, and they have some more options about what tissues they'll differentiate into. I'll get into differentiation a bit more later. And there are a number of ways we can do this, and not all of them technically result in a GMO. Early methods of producing iPSCs involved lentiviruses or retroviruses to integrate desired DNA sequences into the genome of the host cell. Another option is a DNA transposon, which also integrates DNA into the host cell genome, but with the benefit that you can get rid of it once we're done with it. And now, because those are integrative or involve directly messing with the host cell genome, they are GMOs. So more favorable approaches are non-integrative, meaning they don't directly mess with the host cell's genome. For example, we have things like Sendai virus, which will stay in the cytoplasm where they'll replicate for a while and produce factors which encourage the host cell to start to call on its stem genes, which would otherwise be dormant at this stage. We can also do this with mRNA, which codes for the same factors. But in that case, because mRNA isn't self-replicating, we have to do it again and again and again and again and again until the effect kicks in. How many times did I animate that? Okay, good, done. Scaffold building. Now, scaffolds in this context might be a new term for some of you. To use an analogy, in construction, we don't just drop bricks in a pile and expect them to assemble themselves into a building. And similarly, we don't just lump cells together and expect them to assemble into something complex like living tissue. They usually need a framework on which they can adhere and do their work. And if you're wondering why this scaffold is covered in fried eggs, it's because ChatGPT thinks that's what cells look like. There are some cells that can grow in suspension, i.e. without a scaffold, but for our purposes, we'll almost always be working with quote unquote adherent cells, which means cells that need something to stick to. And scaffolds can come in many forms, such as particles, fiber matrices, or hydrogels. But more on that later. Now, for our purposes in cultivated meat, ideally the scaffold needs to be something edible, because if we're growing meat on it and around it, we're either going to have to remove it, which will be very complicated, or we want to be able to eat it with the product. Now we come to media formulation, media being the liquid in which we maintain and grow our cells. And it's basically a potion of nutrients, amino acids, salts, and growth factors. In its earliest form in the late 1800s, it was basically a soup of water and a few different salts and was able to maintain cells for a while outside the body. Just let them survive, nothing more. But then in 1907, someone added frog lymph fluid to the mix, and that enabled the cells to not only survive, but also to grow and divide. Why frog lymph? I have no idea, and I'm not sure anyone at the time did either. But this set a precedent of extracting fluids from animals and adding it to these broths, which continues to this day. It's called serum. And there are various different types, horse serum, goat serum, but the most common type of serum used in culture media is fetal bovine serum, or FBS. And as the name implies, it's a bit gruesome to obtain and kind of incompatible with the aims of cultivated meat. It's also the most expensive component of culture media, which is why current prices of cultured meat are very, very high. All of which is why there are efforts being made to replace it. Serum-free media is an entire field in itself. Now, you might be thinking, can't you just make a checklist of all the different things in serum and mix it up yourselves? Well, serum is chemically undefined, which is a fancy way of saying we don't have a set recipe for it. We know a lot about what's in it, but the exact the exact contents and concentrations of all the different components vary from batch to batch and animal to animal. The FBS you get from one cow will be different than the FBS you get from another cow, and that makes it very hard to replicate and replace. Don't know why I put that in. That's something we would add later. So yeah, there's other things in media like drugs we might add to signal the cells to do certain things we want them to do. It's a very complicated affair. So this is why we have entire biotech companies dedicated to the serum-free media components like Maltus, for example, who make food-grade serum-free media components for cultivated meat applications. And then there's bioreactor design. We can't do everything we need to do in simple culture flasks, so a bioreactor gives us the control we need over factors like pH, temperature, dissolved oxygen, media mixing, and the like. This one is called a dust box, for example, and it can handle 4 stirred tank vessels at once and gave me no end of headaches earlier this year. Bioreactors come in many different configurations, stirred tank being the most common and used a lot in fermentation processes. They can come as large as 50,000 litres, which is the size of a multi-storey building. As the name implies, it's a big tank with an impeller to stir the contents. On the left here, we see a continuous feed design where the media is constantly cycled through. And on the right is a batch fed design where you would manually change a certain amount of media at regular intervals. Another example is an airlift bioreactor, where the media is agitated or mixed by pumping air through it, which is a bit more gentle than stirring if you've got sensitive shear sensitive cells or a delicate scaffold you're using. And some bioreactors are designed to work hand in hand with specific scaffolds, like this hollow fiber bioreactor system, which is also quite popular in the cultivated meat sector. So now we have all the puzzle pieces in place, let's explore a couple of hypothetical models for cultivated meat using the mighty Dodo and the humble T-Rex. Now, for the purposes of this, we're going to pretend that regulations don't exist and that we have no problem with de-extinctionating or GMO making or whatever else, and we're just going to have fun with it from a purely biochemical engineering standpoint. So we'll start with the Dodo nuggets. Now, as you're no doubt aware, the Dodo went extinct about 350 years ago, which means the DNA we have left in preserved samples is quite old, which means it's quite degraded. But fortunately for us, we are not trying to clone a whole living Dodo, for which we would need the entire genome intact. So we can make do with just a small bit of the genome, namely the myogenic genes, myogenic meaning muscle producing. So let's say we either get the myogenic codes we need from a single preserved sample, there's one in the Oxford Museum, I believe, or we could piece it together from many different samples. Excuse me. Completely lost my place. Yes, now we need a cell. So let's say we take a muscle biopsy from a modern bird like a pigeon, and we isolate some myosatellite cells from it as we saw earlier. Test who's paying attention. What we can do then is make use of CRISPR technology, which stands for Clustered Regularly Interspaced Palindromic Repeats, which you may have heard of. Fortunately, you do not need to remember or understand that name to understand the basics of what it does. It's a technology adapted from the immune defense system of bacterial cells, and it lets us take these Cas9 proteins, which are like molecular scissors, and use guide RNA sequences to aim them at very specific DNA sites. So we can cut DNA exactly where we want to edit, remove, or replace certain genes. So we're going to do that to take out the cell's native myogenic genes and replace it with our Dodo myogenic genes. So now we have a cell which is still a pigeon cell, but codes for Dodo muscle. But you didn't think it'd be that easy, did you? No, there's lots more, because even though we have a myoblast which is coded for Dodo muscle, it is not enough to just grow lots of cells. Lots of myogenic cells does not muscle tissue make. Once we've grown enough cells in the expansion phase, we have to differentiate them into mature muscle. And in the case of muscle, this involves fusion. These muscle cells will all fuse together into long tubes, aptly named myotubes, which are multinucleated. So if you look, you can see all the little dark spots lined up along those fibers, which are the nuclei of all the cells that fuse together to make them. And this is primarily done by adjusting the media formulation. A reduction of the serum content, or serum replacement in this case, is the main difference between media optimized for proliferation or differentiation versus proliferation. But there are lots more components to media, and each of them can push cells in one direction or the other towards proliferation or differentiation. Fibroblast growth factor 2, for example, will stimulate proliferation and usually inhibit differentiation, or at least inhibit the fusion aspect. Things like epidermal growth factor will nudge cells more towards differentiation, and things like insulin or insulin-like growth factors can do both. But we can, for example, under hypoxic conditions, which means reducing the amount of oxygen available, insulin growth factors will nudge cells more in the proliferation direction. And that's a modification that will come in at the bioreactor stage. So you can see how the bioreactor works with the media components, works with the scaffolds, and it's all connected. But yes, we'll likely need to develop formulae for proliferation and differentiation separately. And since no one has ever cultured dodo muscle before, we need to optimize them both for dodo muscle specifically. Since different media formulae will affect different species muscle tissue differently. Thankfully, progress is being made in this area. The rise of AI, for example, is letting researchers take their data and use techniques like Bayesian optimization to screen lots of combinations of components at once and zero in on the most promising ones more quickly. Now, something that makes our job a little easier here is that nuggets are not what we call a structured product. Things like nuggets or burger patties, if you've ever watched a Jamie Oliver documentary, you'll know aren't made from whole cuts of meat, rather they're a mass of meat that's been ground up and mushed together. Now, for us, this means we don't have to grow a full muscle. We can grow lots of little bits of muscle and mush it together. And particulate scaffolds are perfect for that. We call them microcarriers. Here you can see an example of some edible microcarriers from Petra's company Quest, with cells happily growing on them, just for illustrative purposes. And there are a few different ways of making microcarriers. I'll quickly walk you through two examples that I've had experience with. First, we have there we are, emulsification precipitation, where you have your microcarrier material dissolved in a liquid, and you mix that vigorously with another liquid in which the material will not dissolve, so a non-solvent. You need to optimize things like the concentration of the material, the amounts of each liquid you're using, and the stirring speed, but this causes your material solution to form spherical droplets suspended in the non-solvent. If you've ever tried to mix oil and water, you'll have an idea of what I'm talking about. Then you can treat the mixed solution in some way to cross-link or otherwise precipitate the scaffold material. If it was silk, for example, you just add some ethanol and that causes it to cross-link and solidify, and then it won't redissolve. And voila, you have some solid microspheres of your scaffold material ready to seed cells onto. Another way we can do this is microfluidic flow focusing, and this also involves the interactions between a solvent and a non-solvent. We pump both through a T-shaped junction, and we pump the non-solvent faster than we pump the material solution. And again, we treat the mixture afterward to precipitate these droplets that form into solid spheres. Microfluidic flow focusing is good because it gives us more control over the size of the particles, giving us what we call a smaller size distribution, which is good. Emulsification precipitation, on the other hand, gives us a larger size distribution, but it's better for making large quantities of microcarriers in less time, which is what we'd want for an industrial application like cultivated meat and making dodo nuggets. So as the microcarrier scaffold goes beautifully with the tried and tested stir tank bioreactor design. As I said, these can come in a variety of sizes, from the size of a water bottle to the size of a building. This makes them ideal for mass production. So one thing we want to bear in mind for our dodo nugget model is that avian cells typically prefer a slightly higher temperature than the standard 37 degrees Celsius that we use in culture of most other mammalian cells. So let's quickly recap our dodo nugget. We start by extracting the myogenic genes from the preserved dodo samples. We don't need the whole genome. We take the modern bird cell, use CRISPR technology to replace the native myogenic cells with the dodos. So now it codes for dodo muscles. And we'll also have to apply some reprogramming techniques to make sure they don't stop growing too quickly. We make lots and lots of microcarriers from an edible material using emulsification precipitation, and we seed them with our reprogrammed cells, the pigeon cells that are now coding for dodo muscle, and we culture them in a stirred tank bioreactor, first using our AI-optimized proliferation media to expand the cells, then switching over to our differentiation media to induce fusion and the formation of myotubes. And then we can squash it all together when it's done to make a mass of muscle mush, which we can then mold and deep fry into some dodo nuggets. And that is more or less the dodo nugget model. So let's move on now to scenario 2 and see if we can make some T-Rex legs. Now, this is going to be a fair bit more complicated, so I'll be giving you more questions and challenges than answers, but hopefully that sparks your imagination a bit. So as we mentioned, DNA degrades. Since the dodo went extinct about 350 years ago, its DNA isn't in the best shape. Now, because of the half-life of DNA, it can be expected to entirely degrade to the point where there's nothing left after about 7 million years, which is a problem for us because dinosaurs went extinct about 65 million years ago. Contrary to what Jurassic Park would have us believe, there is not a single base pair of dinosaur DNA left. But we're not going to let a little thing like apocalyptic mass extinction stop us. If we can't genetically engineer T-Rex meat like we did with the Dodo, let's see how close we can get if we use our imaginations. So a good place to start here might be in 3D printing an anatomically correct T-Rex bone for us to culture cells on. So even if it's not genetically T-Rex meat, it is a structural replica. And to make it as good as possible, we can think about what we can guess about T-Rex muscle. It likely had a much higher ratio of fast twitch fibers than slow twitch fibers, as would be consistent with ambush predators for sudden powerful bursts of activity. The muscle fibers would have been likely very large and thick, unlike modern birds, which are more thin and spindly in terms of muscle fibers. And again, like most predators, it would have likely been very lean without that much fat content, a bit more fat content than modern birds, but yeah, as predators go, they generally have quite lean muscle. So to replicate that, let's try mixing a couple of species, and we'll go with large birds like the ostrich or the emu, which are direct descendants of the T-Rex, and the alligator, which is more of a distant cousin of the T-Rex, but will give us an element of that ancient, thicker muscle structure that we might be looking for. So we'll take someone from our research team, whoever's our least favorite, and we'll give them the task of collecting a muscle biopsy sample from an ostrich and an alligator for us to isolate some cells. But this time we're after more than just myosatellite cells, which brings us to our first challenge. If poking an ostrich and an alligator wasn't enough, that's culturing different types of cells together, or co-culture, as we call it. Different types of cells will have different requirements, and what works well for one might cause a problem for the other. And this extends beyond just that we're trying to culture ostrich and alligator meat together, but this time we're trying to create a structured product. Unlike our last example, where we just needed a mass of muscle mush, this time we're trying to grow a proper muscle, which means we don't just need muscle tissue, but we need adipocytes for fat as well. We need fibroblasts for connective tissue. We need endothelial cells for vascularization. We need lots, and we need to make it all happen together, which makes this model much more complex. It's also a challenge for us in terms of scaffolds to make a structured product like this, because we're not going to be able to grow them in lots of separate bits because we want, proper layers and proper formation of the muscle. So microcarriers are not going to be the best choice. So let's take this in a more fibrous direction. Why not hydrogels? Because I have more experience with fibers, so we're doing fibers. There are various methods of producing fibers, like electrospinning, where the scaffold material is dissolved in a solvent, which is pumped out through a needle to which voltage is applied, and nearby is a grounded collector. And the electrical charge causes the solvent to evaporate and the material to be drawn out toward the collector, like the charge in storm clouds being drawn to the ground as lightning. And this process pulls the material into a very thin fiber. We've also got wet spinning, which involves extruding the material solution into a bath of non-solvent. As the solvent solution dissolves and is replaced by the non-solvent, the material precipitates into a fiber. And then we've got dry spinning, which involves blasting the material solution coming out of the needle with heat so that the solvent evaporates, leaving a solid fiber material behind. So we could apply this here, by electrospinning a fiber matrix around our 3D printed bone, kind of like candy floss on a stick. And then we can culture our alligator and dodo muscle structure in and amongst this fiber matrix to make our muscle. So here's an example of some electrospun fiber matrices from my PhD work. This is made from zein, which is a protein found in sweet corn, which means it's edible. These images are taken using electron microscopy, so these fibers are very small, getting into the nanoscale territory, which is good for replicating the extracellular matrix on which cells tend to grow in the body. Note that the fibers here are all going in different directions. That will be important in a moment. Because another challenge we have with a structured product like this is the need for alignment, at least where muscle cells are concerned. So as you no doubt already know, muscles in the body are striated. All the fibers align in a single direction Yes, like that. And it's the contraction along this single direction which lets the muscles function. In a Dodo nugget model, this wasn't really a concern. We could just let them settle whichever way they wanted, which is the default for cells. When you see them on a surface, they'll align themselves randomly. But for a structured product, we need to sort that out. Now, one way we can do that is by aligning the fibers themselves. If you remember that previous image I showed you, and we said the fibers weren't aligned, when you're electrospinning them just onto a collector, they'll align themselves in random orientations. But what we can do in this example is rotate the collector, in this case our 3D printed T-Rex leg, as we're spinning fibers onto it. And that will result in the fibers all being pulled into a consistent alignment. And that in turn will create a topography which help the cells figure out which way they should be pointing. Additional ways we can induce alignment and help our product along at the same time is by stretching it and exercising it. So when culturing muscle tissue, we can apply electrical stimuli to induce contraction, and we can manually stretch the construct, both of which aid in the development of the muscle tissue and depend on the myotubes being aligned properly. But I'm not done throwing problems at you yet. Our choice of scaffold limits us with regard to what kind of bioreactor we can use. If we try to put a fiber matrix or a block of hydrogel into a stir tank, the impeller will just destroy our construct. So we need to invent an entirely new purpose-built bioreactor design into which we can put our candy floss dinosaur bone and seed our ostrich and alligator cells onto it. This time we need to optimize our media for differentiation of multiple cell types at once. We need to figure out ways of integrating, ways to stretch and zap the construct. It becomes a huge engineering challenge, which is why we need plenty of talented bioengineers. Like I said, more questions than answers for you for the T-Rex model. Hopefully that's fired up your inner engineer a bit and sparked your imaginations. So, whoops, what did I just do? There we go. So to wrap up a bit, let's step back and have a look where cultivated meat is now. So when I joined the field, a friend gave me this in vitro meat, which you can't see because I blurred the background, in vitro meat cookbook. Shout out to you, Hannah. I recommend checking it out if you've enjoyed these kinds of hypothetical cultivated meat models, because it has a lot of those. One thing this book posited back then was cultivated pet food, which has now reached the UK market in the form of cultivated chicken dog treats from a company called Meatly. Now, I don't have a dog, so I haven't had the occasion to use them, but I do know people who have. Alice Johnson here, for example, from Aston University, has given them to her dog Coco. Keep your eye on Alice, she's going places. Japan's Integraculture have recently produced cultivated chicken nuggets and cultivated duck liver. We have been dealing with some pushback. Florida, for example, outright banned cultivated meat, calling it a plot by the global elite, although there is an ongoing federal lawsuit to overturn that. And Singapore is kind of the hub of cultured meat innovation because the regulatory approval is a lot further along over there. A lot of companies are looking to trial their products out there, and there is some cultivated meat available to purchase for public consumption in Singapore. So hopefully that has given you a quick look into cultivated meat, where we are now and how far we have to go. Just put my email there in case I don't get around to all the questions, because I hope you get a lot of them. So if I don't get around to your question, you want to email it to me, feel free. So thank you so much for your curiosity and your attention. And I hope this has sparked some new ideas for your own journeys ahead. And that's all from me.
Great, thank you very much indeed. So what I'm going to do is so if you'd like, please post your questions using the Q&A box. You'll see at the bottom of your screen there. Whilst I'm waiting for people to answer, I've also just want to say, if we've answered absolutely everything you want to know about the subject, and we've set aside time in the session to answer your questions. But if we've come to the end of them, thank you. It's a very nice comment there in the chat. Thank you. Yeah, yes, he's an awesome presenter. So yes, but if we get through your questions, we're more than we've set this time aside, but we don't have to keep going. So we'll keep answering your questions as long as they're coming through. So whilst we're waiting for any questions to come through, I want to shamelessly plug the fact that we also have some more of these virtual taste select sessions coming up. So we're going to be doing one on vaccine manufacture. We're also doing one with Fernando. He's going to be talking about the challenges of transporting medicines around the world, which is something which was really critical during the pandemic, about how you can have a vaccine which is a solution to a global health problem, but if it has to be transported at minus 80 degrees centigrade, it's not a solution for the whole world. So trying to make vaccines, trying to make medicines easy to transport, cheaper, it means that they potentially reach a much greater percentage of the global population, and they represent a much more significant health solution. So those we've got those coming up for the rest of the week. I will post links to those in the chat. But I also wanted to say, if you're interested in doing a degree with us, I will be joined online by Dr. Jack Jeffries. He is the admissions tutor for our degree programs. And on the 5th and 6th, he will be online with me, and we will be we'll be able to answer any questions you might have about our degree programs. If you're interested in doing an MSc, and I noticed that a couple of people might be interested in doing prospective MScs, what I'll do is when we send a follow-up to this, I'll make sure that you can register with UCL. And when we have our next sessions coming up for MSc manufacturing commercialization of stem cell and gene therapies, the synthetic biology MRes, and the program that's probably most allied to the work that Travis has done, which is the MSc in Biochemical Engineering, which is actually led by Dr. Petra Hanger. When we have some sessions coming up where you can talk to people involved in those, the tutors, the admissions tutors, so you can have a chance to do that. So I see we've got a few questions come in. What I'm going to do is, because I've noticed this has come through on the chat, I'm going to ask this first of all to Travis, because it's such a good question. And it does come back to something Travis said at the start, but I think it also, I'm going to ask you to expand on it a little bit, which is, what inspires you most to work within the cultivated meat industry? And also, what first of all brought you to biochemical engineering? So two sort of sides of that, because I think it's quite useful to know why people came into this sector in the first instance.
Can you hear me?
Sorry, yeah, you just broke up for a little bit there. If you could just
Sorry.
repeat what you said there.
Yeah. Am I back? Cool. So what inspired me to get into the sector? For me, it was mainly driven by animal welfare. I'm a big animal lover. I like animals more than people, I think. So I wanted to see how I can combine my passion for science with animal welfare. And that led me to the thought, oh, what if we could grow meat in labs rather than slaughtering animals for it? And then I found that other people had that idea already. Back then, the field was very, very small. So I think when I joined the company in Cambridge, I was one of 5 people. Last year, I attended an event called Cultivate, which was like this time in a proper lecture hall, like 100 attendees. But I remember going to the very first Cultivate conference, which was 12 of us in this old church hall sitting around a heater because the heating didn't work, all shivering. Where was I going with that? Yeah, so mainly animal welfare from me. I think the majority of people I've worked with are into it from an environmental perspective. I haven't really encountered very many fellow vegans and vegetarians in the sector. There are a few, but not that many. What was the rest of the question, Kim? Sorry, you had an expansion on it there.
So no, I mean that I guess actually that those are the same things. So why you got into this in the first place, and what inspires you. So I mean it's interesting that if you ask the same question of Dale and Pierre, who were there with us yesterday, they would have, I mean, they were talking about very specifically stem cell and gene therapies, which is what inspired them to get into the field of biochemical engineering. And and also I think if you ask somebody what is a biochemical engineer, Travis is a biochemical engineer, Pierre is a biochemical engineer, Petra is a biochemical engineer, someone like Professor Martina Micheletti, they are biochemical engineers, and they are addressing a global problem, but using the tools that a biochemical engineer has available to them, which are essentially the bioprocessors. These are the amazing tools that you can use to create the materials, the foodstuffs, the medicines of the future. Sorry, I get very excited about this stuff, having worked in the field for a while. One quick question, which I'm going to come to you now. What's the name of the book title that you mentioned towards the end there? If you
If I just quickly unblur my screen so you can see it, that is the In Vitro Meat Cookbook. I believe that was made by Isha Dattar from New Harvest, which is a cultivated meat funding body. Yeah, the In Vitro Meat Cookbook. Worth a read, I would say. Let's see, we've got two other questions. So we've got, can you biochemically engineer lab-grown meat to alter its vulnerability to diseases or bacteria like salmonella? I would say we can. And there is kind of research and thought into doing that, as well as things like enhancing the nutrition profile. The problem with it is it would involve things like the GMO techniques. And when you get to GMOs, there's a lot of regulations in the way about GMOs for food. Like in the EU, for example, it's very tightly regulated. And if you have a GMO product, it's going to be difficult to get it to market. So whilst it is theoretically possible, it would throw up a whole load of barriers in getting the product out there. Hopefully that answers the question. And we've got, think of me here, given how cultivated meat species is growing. Oh, Kim would like to answer this question. Okay. With companies like Wow creating entirely new products like quail eggs and even mammoth meatballs, while others like Mosa meat and higher steaks are sticking to more familiar meats like beef and pork, what do you think is the better strategy for gaining public acceptance? Should companies focus on reinventing the menu with novel products or lean into the familiarity of traditional meats? My personal, sorry, I nudged the microphone. My personal view on that is that the latter would be better. Stick to the traditional things. People are a bit wary of new technologies, especially when it comes to something that they're going to be ingesting. A lot of the public concern around this space is in things like transparency. They want to really understand what goes into this product. So I think further down the line, it could be good to bring out some novel products. But I think for getting cultivated meats solidly into the market, these traditional meats like minced pork or burger patties will be the way to go. That's my personal take on it. Although Singapore being the kind of hub of cultivated meats, tend to be more open to these new experimental technologies. So maybe in that demographic, they might be more open to novel products. But I think globally, keeping it simple, at least to start with, is the way to go.
Fantastic. I just want to mention here that we did a Spring to STEM, which was a live session, which was there was a talk given by my colleague Farnaheen Marslan. She was focusing very specifically on medicines that were created without actually using animal products because they were not halal. And this is an area where there are many people who cannot accept medicines that are created if they use products from pigs, and this means there are many people who are not having life-saving therapies. So again, this is another approach where you talking where the ethics and the morality, but also the practical application of biochemical engineering becomes really critical. It also starts to become, sometimes it's as critical to look at the regulatory framework in which you operate as well as the scientific framework. And I know that is it William is very interested in the regulatory framework. So again, if you can influence policy, you can sometimes have as much and sometimes more impact than you can in a laboratory. They have to work together. Somebody's actually asked if there's going to be a recording available of this session. Absolutely, yes, there is. It will take a while for me to upload all of them. I also need to, there's a bit of processing involved, so it'll be after next week, but all of these will be available to watch afterwards. I'll also make sure that it's shared via Zoom as well. So if you're in a part of the world where you can't watch on YouTube, that'll be available to you. So there are a few more questions that have come through, so I'm going to hand over to
Yeah.
you first.
Sitting working in your lab during RBS, is it limited to, well, I don't know the specific admissions policies, but I will say I don't have a degree in biochemical engineering. I did my undergrad in biomedical sciences. Then my PhD was technically in pharmacy and pharmacology, not because it had anything to do with pharmacy and pharmacology, but that was just a department that my supervisor was in. So yeah, I don't let things like that limit what you're exploring doing, I would say.
Also, having had the privilege of being in MSc Biochemical Engineering sessions with Petra, um, one of the unique aspects of our MSc in Biochemical Engineering is that it has streams for biochemical engineers, chemical engineers, and life scientists. So it's got tailored programs that will actually take you towards being an accredited biochemical engineer. So if it's something you're really interested in doing, and your background is in life sciences, we've had people coming from biomolecular science. So there are many routes in for the MSc in biochemical engineering. If you'd like to know more about that, please get in touch with me directly, and I'll be more than happy to send you details of the different routes in for different kinds of scientists.
And we've got another one. So most of the products, at least initially, will be unstructured products. How far is the research for structured products? Ah, now it's a tricky one to answer because a kind of a problem or a barrier in the space is that a lot of the research is done in industry, and all these companies need to protect their IP, which means there's not a lot of information exchange, uh, at least outside of conferences. So it's kind of hard to say how the research into structured products is coming along. It's I think the main point I was making with my part is there's a lot of challenges to overcome. So I don't think we are getting there yet. Although that being said, the chicken nuggets that Integriculture have just come out with, they are reporting to be structured chicken products, and they've made those using hollow fiber bioreactors. But because it's a commercial product and they've got their IP protected, there's no way I can really say what's gone into it or how they've made it. So it's a bit of a tricky one to comment on, but hopefully that gave you something. We've got, you mentioned a stirred tank bioreactor isn't really suitable for growing meat on fiber scaffolds. What type of bioreactor would you use instead? Yeah, I forgot to say that, didn't I? In that particular example, since we will have like a long bone-like collector with fibers on it, I would think something like an airlift bioreactor would be ideal, or maybe a rotating wall vessel, something that will be quite gentle with agitating the media, because these are quite shear sensitive cells as well, and the scaffold is quite fragile. I can tell you from working with electrospun matrices, they come apart and degrade a little too easily. We got, you mentioned, I answered that one. Was there another one that I missed?
No, there was just somebody was asking. There was somebody asking details about different routes in for our programs. The best thing to do is get in touch with me directly. I'll make sure that we'll invite you to one of the webinars where Dr. Petra Hanger will be able to talk to you in full detail about the program, discuss the different routes in. Also, it's if you've got a degree in a related subject, we kind of need to see the exact modules that go into that. We're best having that conversation offline, and I can put you in touch with the correct people. So I've just sent you my email address on that one. So I think we've answered all the questions that were there. Yep, there's one more that's come in, so I'll clear these ones out of the way, and I'll leave the last one for you to answer, Travis.
So we've got, wonder what the growth media hormones are used in culturing meats. So it depends on the species of animal you're doing, and also what types of cells you're wanting to cultivate. Usually things like FGF, insulin-like growth factors are quite standard, but there are a lot of these that we need to find replacements for because the media needs to be food grade. So that's why we've got companies like Maltus who are producing these components or analogous components that are serum free and food grade. Again, there are companies, so they don't really disclose all the details of how they make their product, otherwise everyone would steal their idea. So a lot of these growth factors are active in humans, but they are species specific. So the growth factors you'll find in one species will be slightly different, quite similar, but slightly different to one that you'll find in another species. So in cell culture in the lab, often we will use human FGF for research purposes, but for the purposes of a cultured meat product, we want to find something else that can be food grade. Dexamethasone, yeah, that's different from the serum and the growth factors. I think I was going in a different direction when I was putting that slide together. So dexamethasone is a drug, and it's used a lot to stimulate muscle cell proliferation, but also adipogenic differentiation, so getting cells to convert to fat cells. It's kind of a gold standard component in adipogenesis in the lab, but because it's a drug, again, we need to find something else to use in place of it in the culture media. Hopefully that answers the question a bit.
It's just occurred to me that we hosted a conference last year on lab grown meat, and a couple of PhD students actually wrote it up. So I'm going to put a link to the article we did because there's some really interesting stuff in there. And also, given the interest in this webinar, you might want to sign up and come along to UCL East and maybe join the next one. I know that Petra is hoping to do another one. It was a very interesting session, really, really good panelist session. So I'm going to pop a link to that in the chat here. And so you can actually, if you want to read the article, that's a fascinating story. That was from May last year, so it may be that we're going to be doing that soon. Anyway, so I think those two questions, I'm just going to clear those out. So I think that we've covered everything there. So I think to wrap up, I just want to thank you very much for coming along and joining us today, and for your questions and your interest in the subject. It's been great to have you on this Tuesday morning. Huge thank you to Travis for taking the time to give this talk. That was great. I'm very lucky I get to sit in on all of these and enjoy them. It's always a great privilege. If you please, I'll send you a link afterwards to make sure if you've not signed up through UCL, do so, because it means that we'll be able to tell you about other things that we have coming up. And also, if Travis, if you're happy, I'll give your email address if you want to get in touch with you. And do come along to the next lab cultured meat conference that we're doing in UCL East. It was really, really good last year. But that's it. So thank you very much for coming along today. And I hope you have a great week. And I hope to see I hope to see a lot of you tomorrow and the following days for the next virtual tasting lectures. Thank you very much. Have a great day.